Hi guys. So today I'm going to finish up the lecture first talking about neurotransmitters, and then I'm going to talk about how different kinds of drugs affect our neurotransmitter systems. So the last time we left off, we talked about what happened at the very end of an action potential. So an action potential is the firing of a neuron in your brain. It is what happens when you have any kind of experience in the world, any thought, any feeling, the beat of your heart, every light you see, every sound you hear, everything that is your experience is a product of a symphony of action potentials. And at the end of an action potential, the very last act of an action potential is to release the neurotransmitter from the neuron. And then that neurotransmitter is going to travel across the synapse and bind to a receptor somewhere. Well, there are a lot of different kinds of receptors. There are a lot of different kinds of neurotransmitters. And so we need to understand something about those different neurotransmitters so that we can understand how they work in our brain, the different kinds of things that they can do and control, and how different substances that we use might affect those neurotransmitter systems, and ultimately then, of course, affect our behavior, affect our thoughts, affect our feelings, and affect what we do. So I did talk a little bit last time, but I'm going to review it very briefly again. I talked about GABA, glutamate, and glycine, the amino acid neurotransmitters, but I'm going to just mention them again briefly. And then I'm going to move on, talk about acetylcholine and what acetylcholine does in our brains and in our bodies. Acetylcholine is the one that's abbreviated ACH. We're going to talk about acetylcholine in the peripheral nervous system and in the central nervous system, because it does different things in those different places. And then I'm going to talk about the monoamine neurotransmitters, and there's a few of those that are very famous. The catecholamines like dopamine and norepinephrine are very well known, and we'll talk about why. And serotonin is the final monoamine neurotransmitter I'm going to talk about. And if you put A, B, and C all together, the amino acid neurotransmitters, acetylcholine, and the monoamine neurotransmitters, your book calls those the small molecule neurotransmitters. So those are what I'm going to talk about first. Okay, so hopefully this very first part is just review, because I did mention it last time. GABA, or gamma aminobutyric acid, which you don't ever have to write that out. All I want you to know is GABA, okay? GABA is the major inhibitory neurotransmitter in your brain. When GABA is released by a neuron and travels across the synapse to bind to a docking site on another neuron, that docking site is on a protein, and that protein is a receptor for that neurotransmitter. And so GABA, little molecule GABA, comes across and it docks to the binding site on the GABA complex, this GABA receptor right here. And when it does that, GABA receptors are ionotropic. It immediately opens an ion channel. And GABA, being the main inhibitory neurotransmitter in your brain, is going to let negative ions into the postsynaptic cell. The negative ion that it's most likely to let in is chloride. Because remember, all in that extracellular fluid is sodium and chloride, salt in the salt water of your body. And the thing that comes in through a GABA receptor, when a GABA receptor receives GABA, GABA binds to its docking site, is chloride. And that is the major way you inhibit cells in your brain. Most of the cells in your brain have GABA receptors on them. Most of the neurons in your brain have GABA receptors on them. And when GABA does its job and binds to the receptor site, it's going to let chloride in, and that is going to hyperpolarize this cell. It's going to send this postsynaptic cell more negative, further away from action potential. It is going to make it less likely that this postsynaptic cell fires. Glutamate, I mentioned before, is kind of the opposite of GABA. Glutamate is the main excitatory neurotransmitter in your brain. And so when glutamate binds to a glutamate receptor, generally speaking, that's going to open an ion channel that lets in a positive ion. And usually that's going to be sodium. So when glutamate binds to its docking site, it's going to open an ion channel. The ion channel is going to be for a positive ion, usually sodium. Sodium is going to come into that postsynaptic cell, and that is going to depolarize the cell. That is going to push it towards action potential. It's going to be going up from that resting potential of negative 70 millivolts towards the threshold potential of negative 55 millivolts. Okay, so that's the job of glutamate, is to excite cells. And your brain also has glutamate receptors all over it. Those two neurotransmitters can be thought of as the workhorses of your brain, doing the major excitation and the major inhibition that needs to be done, that creates you, that creates your experience of everything you think or feel or sense in any way, everything you do. All of that is controlled in large part by glutamate turning brain cells on and GABA turning brain cells off. And it's very important to do both, because if you didn't inhibit brain cells, you would be having a seizure all the time. Too much activity of your brain cells doesn't give you cool superpowers, it gives you a seizure. So you have to have a way of deactivating the neurons you don't want to be using at any given time and activating the ones you do want to use. You know, sometimes you want to drive a car, sometimes you want to walk through a park, sometimes you want to write a paper. And you're using different neurons for all of those things. So you want to turn off the ones that are not right to use, turn on the ones that are right to use, and that's mostly GABA and glutamate doing that job for you. I'm just going to mention glycine very briefly. Glycine is inhibitory like GABA is, but glycine is found mostly in your brainstem and in your spinal cord. So probably what happened is we evolved to use glycine as an inhibitory neurotransmitter first, but once we evolved to use GABA, it was more efficient and flexible in the way we could use it, so we started using GABA as the major inhibitory neurotransmitter for the rest of our brain. But you do see glycine still in those areas of our brain and used in lower order animals. But now I want to move on to acetylcholine. So acetylcholine is abbreviated ACH, and acetylcholine is an extremely important neurotransmitter in our brain. It is not just something we find in the peripheral nervous system, right? So that would be your cranial nerves and your spinal nerves. It's also something we find in the central nervous system in our brain and in our spinal cord. Which do I talk about first? Okay, I'm going to talk about the peripheral nervous system ones first. Acetylcholine producing cells, neurons that produce and release acetylcholine at the end of action potential, are found in a very important place all over your body. And that is at the neuromuscular junction. You see it written right there. Take apart that word, neuromuscular. Neuro has to do with the neuron. Muscular has to do with the muscles. You know in this class that it's your brain that controls your muscles, whether you're talking about your voluntary muscles or your involuntary muscles. So your muscles in your arms and legs or your muscles in your intestines, okay? Regardless of what you're talking about, that place where the neuron comes together with the muscle is called the neuromuscular junction. And so one of the functions of your neurons is to move you around. How does it do that? Well, you have neurons in your brain and in your spinal cord, and the cell body The gray matter part is in your brain or in your spinal cord, but the axon projects out to all of your muscles. And here is an image of that axon, of that neuron, coming down and projecting to a muscle. Let's say this is a muscle in your arm. So here is the end foot of the neuron filled with neurotransmitter. What neurotransmitter? Acetylcholine, ACH. And at the end of action potential, there's going to be voltage increasing here in the end foot. That's going to bring calcium into the end foot. That's going to cause these vesicles to bind with the cell membrane, and you're going to release neurotransmitter into the synapse. But down here, these receptors that you have, these aren't on another neuron. These are on your muscles. These are receptors on your muscles. You have receptors on your muscles that acetylcholine can bind to. All of the motor neurons that leave your spinal cord release acetylcholine. So this is what happens when you are controlling any kind of a muscular activity you do. It's the same process. So acetylcholine binds with its receptor and it opens up a sodium channel. Sodium channel, sodium is in the extracellular fluid, so sodium comes inside the muscle. And that's what contracts your muscles. Salt comes into your muscles and contracts your muscles. It's the same reason that when you see those videos where the person puts out like frog legs and then they put salt on it, or like they're eating some kind of seafood and they have, like they chop the head off a squid, but they put the tentacles there, and then they put like soy sauce, right, with a lot of salt on it, and it makes the legs start dancing around. Well, it's that same thing, except for instead of releasing acetylcholine and nicely opening some little doors for the sodium to go in, you're just dumping it on the muscle and making it twitch. You know, this one you can use again and again. Usually that one, you know, you do that for a little bit and that's the end of that. Okay. These receptors at your motor end plate, these receptors on your muscles for acetylcholine are called nicotinic receptors. Nicotinic receptors. Nicotinic acetylcholine receptors are excitatory. Nicotinic acetylcholine receptors, when that acetylcholine binds with that receptor, that nicotinic receptor, it opens up a sodium channel and positive sodium ions come in. So that's an excitatory process. That leads to an excitatory postsynaptic potential, an EPSP. This is excitatory action that's going on here. And that can also happen in your central nervous system with acetylcholine. And you might have already guessed that because they're called nicotinic receptors. Well, guess why? Guess what binds with those receptors just like acetylcholine does? Nicotine. That's the first thing we discovered. Nicotine, and we'll come back to this in a little bit, is something that can bind with nicotinic receptors just like acetylcholine can. But we'll come back to that. In your parasympathetic nervous system, okay? So your parasympathetic nervous system, your autonomic nervous system, okay? We're talking about part of your peripheral nervous system, but we're talking about those autonomic nerves. Those nerves that you don't have voluntary control over. Your involuntary nerves, those autonomic nerves. And one part of your autonomic nervous system, you'll recall, is called the sympathetic nervous system, and that controls your fight or flight response. But the other one is your parasympathetic nervous system, your rest and digest response. And remember, when one is on, the other is off. So what's interesting is that acetylcholine is also in charge of controlling your parasympathetic nervous system. There are acetylcholine receptors in your parasympathetic nervous system. So they receive acetylcholine, but they are not nicotinic receptors. They are called muscarinic receptors. And even though muscarinic receptors have the same key as nicotinic receptors, which is acetylcholine, even though they have the same key, so the lock is shaped the same way, the kind of ion channel that's opened is a different one. It's an inhibitory ion channel. It lets in negative ions. So in your parasympathetic nervous system, you have acetylcholine receptors, but they are muscarinic acetylcholine receptors. When acetylcholine binds there, it turns down the activity of the secondary cell. And that sounds a lot like what happens when your parasympathetic nervous system is doing its job. It turns down your heart rate. It turns down your breathing rate. It slows things down in your body so that you can rest and digest. So acetylcholine is very important for doing both jobs, but in one case it's going to do an excitatory job, create EPSPs, and in the other case it's going to do an inhibitory job and create IPSPs. There are many acetylcholine pathways in the brain, and your acetylcholine pathways are heavily involved in your explicit memory. So your memories of facts and events, what you probably think of as your main memories, acetylcholine neurons are active when you are having one of those memories. So for example, your hippocampus, that's where I mentioned that Alzheimer's disease is going to attack the acetylcholine neurons. Okay? So I mentioned that in class last time. Your hippocampus, full of acetylcholine using neurons, a lot of nicotinic receptors, really ubiquitous all across the brain. And those are the ones that are going to be attacked by Alzheimer's disease. And so it is in allowing them to function better, the ones that are left, that we can help people to retain useful memory for as long as possible in the Alzheimer's process. Of course, it is degenerative, so eventually they are going to lose that ability and not be able to remember much of anything at all. Okay. You do have acetylcholine receptors throughout the brain, basal ganglia too, yes, and all over your cortex. So I don't want to make it sound like it's just in the hippocampus, but that was one of the areas where they're very dense, and we talked about it last time. Okay. Okay, so now I'm going to talk about the monoamine neurotransmitters. And the first little category under monoamine neurotransmitters are the catecholamine neurotransmitters. And the two catecholamine neurotransmitters that I'm going to talk about right now are dopamine and norepinephrine. Okay. Dopamine receptors are mostly metabotropic. they are not ionotropic. So they are more slow acting than ionotropic receptors. Your dopamine receptors, I've mentioned before, are affected in Parkinson's disease. But the way I told you that they were affected in Parkinson's disease before may not have been by saying it was dopamine neurons that were affected. What I probably told you, what I know I told you, is that it's your nigrostriatal pathway that is impacted by dopamine. So remember, in your tegmentum, you have this thing called the substantia nigra, this spot called the substantia nigra, that helps you initiate movement. And the substantia nigra there in your tegmentum has projections that go to your basal ganglia, and a particular part of your basal ganglia called your striatum. And that is your nigrostriatal pathway, okay? All of these pathways are named after where they start and where they go. So the nigrostriatal pathway starts in your substantia nigra and goes to your striatum, which is in your basal ganglia. And that is the pathway that is responsible for your ability to initiate and control a lot of your motor movements. And I've mentioned that it's this pathway that is destroyed by Parkinson's disease. Well, that pathway, the nigrostriatal pathway, is a dopamine pathway. So the kinds of neurons that are being destroyed are dopamine neurons. And so when they're destroyed, you get the characteristic symptoms of Parkinson's disease. Difficulty initiating movement, and muscle rigidity, and difficulty learning new movements. But there are other pathways that use dopamine. Dopamine is not just important in our motor pathways. It's also important in our pathways for pleasure and reward. And we call those our mesolimbic pathways. From our midbrain, that's what meso means, to our limbic system. And our limbic system is kind of like our lower cortex. It's the stuff that's all wrapped around the thalamus. It's pretty primitive areas of our brain. That's where we experience feelings of pleasure and reward, when those areas of our brain are active. Those pathways also extend to the cortex. And so sometimes you'll hear people refer to the mesocortico-limbic pathways. Now those are pathways that go from the midbrain to the cortex and the limbic system. Mesocortico-limbic pathways. So now if you're going to the cortex, you're involving higher order function, higher order thoughts, higher order cognitions. And it is your dopamine system that is over-functioning when you have schizophrenia. Folks who have schizophrenia have an over-functioning, especially mesocortico pathways in their brain, dopamine pathways in their brain. There's a lot of extra use of dopamine in those mesocortico pathways that's happening in schizophrenic folks. It's totally naturally occurring. It is not something that a person has any kind of control over. It is genetic in origin, and then the onset of the disorder, like many disorders, can be triggered by certain events. But what happens is we just have way excess dopamine in that area of the brain, in those mesocortico pathways, dopamine pathways. And so what you get are the characteristic symptoms of schizophrenia, or any psychotic experience. That involves delusions. So delusions are beliefs in things that are not real. So perhaps you think somebody's after you, but nobody's really after you. Or maybe you think the radio's talking directly to you. So those can be beliefs in things that are not true. Or you can have actual hallucinations. So hallucinations are where you see or hear or feel things that you can even smell or taste, things that aren't really there. So you might hear a person talking to you, or it can be anything. And that is completely beyond the person's ability to control. It's over hyper functioning of those dopamine neurons in those pathways that is causing something to happen in your brain as if it's really there. The same neurons are activating as if that person was experiencing that thing as if it were really there. And so, obviously, that's producing an experience that is not in keeping with reality. Okay. Norepinephrine, which is also called noradrenaline, and is usually abbreviated NE, is active in your sympathetic nervous system. So a lot of norepinephrine is being released when you are having excited activities that you need to be up for. And what it probably does is it arouses your brain and central nervous system. We find that people who have depression have reduced levels of norepinephrine neurotransmitter in their brain. And so we know that less of it is associated with depression, more of it is associated with not having depression. And we think that probably norepinephrine allows you to kind of have focus and increased attention, extra alertness. So these are obviously things that you want to have when you're doing things like studying, and these are also things that are pretty much impossible for depressed and anxious people to do. Depressed and anxious people do not have normal levels of norepinephrine, and so they are going to have a much harder time with the focus that is needed for, doing higher order cognitive function and being awake and aware. So those are the catecholamines, but the last monoamine neurotransmitter that I want to mention is serotonin. And serotonin is kind of a famous neurotransmitter. It's found all over your central nervous system and your peripheral nervous system. And there are a lot of serotonin-using neurons in your limbic system. And your limbic system, so it's being this, kind of area wrapped around the thalamus, doing some kind of higher order stuff, but not quite to the area of abstract thought or anything like that. But it does control a lot of your mood. That area is very active in mood behaviors, your feelings, your emotions, your sleep. Okay, your limbic system is heavily implicated in sleep. And so if serotonin pathways are all over your limbic system, then serotonin is going to be important to sleep and mood. And it's definitely one of those neurotransmitters that we see associated with depression, anxiety, and mood disorders. Now I'm going to talk about your peptide neurotransmitters, and then I'm very briefly going to mention your gas neurotransmitters, okay? Mostly I'm going to leave that up to you. Your peptide neurotransmitters include your endogenous opioids. Your endogenous opioids, I'm going to talk about those guys right now. Peptide neurotransmitters are just called peptide neurotransmitters because that's the kind of molecule it is, a peptide molecule. And they are, we call them neuromodulators. What does that mean? It means they modulate other neurons. Okay, so peptide neurotransmitters aren't so much responsible for a lot of their own, in charge of a lot of their own functions, but they are in charge of turning up and turning down other important functions. So, for example, you have a kind of neurotransmitter in your brain called an endogenous opioid. And this is a rat brain where everything that has colors, okay, so whether it's red, yellow, green, blue, or purple, all the colored areas are areas where there are endogenous opioid receptors in a rat brain. So everywhere. And we have these in our brain, also everywhere, endogenous opioid receptors. You have probably heard your endogenous opioids called endorphins. And you may have heard that associated with a thing called a runner's high. So I've actually experienced this. Oh my God, I used to run. Gosh, that's a lifetime ago. Okay, I used to run, and I remember I'd be not sitting there, running. Oh God, I hate this, 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 I hate this. And all of a sudden you get this feeling, ooh, wow, what's that wonderful feeling? I feel like I could run forever. People who are in, marathons and stuff like this will describe this runner's high, this second wind where they feel good again. Well, what has happened is you've released your endogenous opioids in your brain. And your endogenous opioids are associated with pain relief. You are asking your body to do something ridiculous that it shouldn't be doing. You are asking it to run a marathon. And that is absolutely ludicrous. And so your body says, okay, crazy, I am going to release this endogenous opioid, and it is released and it binds to its receptors and it diminishes your pain so that you can keep doing that crazy activity and run the rest of your marathon. Okay. The gas neurotransmitters like nitric oxide, here's what I want to mention about nitric oxide. You should really review the gas neurotransmitters carefully in the book because I'm going to put a test question on there about them. But what I want to mention is that gas neurotransmitters go right through a cell membrane. When we've been talking about all these processes, getting ions in and out, which ones can move back and forth across the cell membrane, we're talking about very small molecules. But gases are even smaller than that. So gases can cross right through the cell membrane. And when they do that, they can affect not just the postsynaptic cell, but they can affect the presynaptic cell and other nearby cells as well. So gas neurotransmitters, we probably don't use them, we don't use them nearly as often as the other things I've been talking about, but when we do, they have a powerful effect. We use them for things like sexual and defensive behaviors to allow our blood to flow very quickly, very easily. Okay, the rest you should look up in the book.